Okay, we're going to do another plan diagram. This time I thought it's only fair that I take something I'm unfamiliar with. In the exams you might be given something that you're not familiar with, and so I thought this video might show you just how to deal with that. So this is a transverse section of a leaf still, so I should be able to label some things, but there are definitely some structures here that look a bit interesting, especially the ones that the black arrows are pointing to. Alright. So let's get into the drawing. So I tend to start from the top. So drawing my epidermis. Getting a little bit close there, but as long as those lines don't touch, that should be fine. Drawing in the next layer. So we've got a cell layer, a layer of about 3 cells, 2-3 cells thick, just underneath there. Our next layer, alright, I know this one, it's the palisade mesophyll. Draw that in. Alright, then we've got spongy. I might go down to the bottom layer. So down the bottom here, we have a layer that's kind of broken or sort of interrupted. So let's go draw maybe okay, and now I can see there's a thin layer of cells that comes in here, and again I probably need to draw that as a double layer. Alright, and especially if we look at the middle here, the middle section, there is actually a thin layer, which is the epidermis again on the other side, which actually continues round in. And I'm guessing on the other side, that outer layer, even though it's very pink, on this side it's not so visible in the picture, but on this side you can see it again. So there is actually, the epidermis does go right around. So I might draw this one in first. Okay, fantastic. So let's draw in the epidermis. Okay, and even though it wasn't so clear on the left, it's definitely something I'd draw in, because it's something I'd definitely expect to actually see in a plant. So those collenchyma, parenchyma sort of layers may not always be so clear. You could probably leave those out, but sort of an epidermis is something that's going to be pretty standard. Same with the palisade and spongy mesophyll. Sometimes it's hard to see where it starts and finishes, but even if you just labeled it mesophyll, more generally just the two layers together. Okay, so in here there are tiny hairs. There are these kind of sort of spiky looking structures that come off. They're maybe not quite so clear. And I'm not really paying too much attention to where each individual one is actually situated. Remember, this is really just supposed to be representative of what is in the middle there. Now, what these are actually is stomatal crypts. they're actually indentations on the underside of a leaf, so the stomata are found in here rather than in the other parts. The stomata are probably not so clear. We can see slight areas where it's darker. They look almost like nuclei, alright, if we were looking at a cell that are sort of around here. Those are probably your guard cells, although it's very difficult to see sort of necessarily a pair of them. It's not that clear. So I'd maybe even just leave it at that. What we can still label is obviously our veins. So I've got sort of veins probably just on the border of our palisade and spongy mesophyll. There's another one over here. So a vein or a vascular bundle. I'd probably maybe erase that bit there. I got a bit heavy or a bit dark where my lines maybe didn't quite meet up so well. I'm just going to skip that for the sake of the video. You don't want to watch me have to erase stuff and do it again. Now you can see there is actually sort of a layer of cells around the edge of the vascular bundle, so I might draw that in. This one around the outside. They're actually known as the bundle sheath. Okay, so this bit I'm not 100% happy with, again, so that I might erase or do again. And then inside here it does kind of split across. Okay, and I think that's probably all I'd include. So in this case, I would label the things I'm definitely sure of or I know. So let's start with the upper layer. So this is my epidermis. This is my palisade mesophyll. This is my spongy mesophyll. Okay, down the bottom here we've got a lower epidermis. Okay, let's come out maybe down to the bottom. We've got here the slightly larger cells on top are our xylem, and these ones would be our phloem. Okay, and that would basically be it. Remember our title, so genus, and then the species, if we know it. If we don't, if it was, like when we do onion cells, you can just write onion cells. If I really didn't know anything about this, I would probably I'd probably do leaf, transverse section of leaf, just so we've got some titles, so someone we know is roughly what we're looking at, and then obviously magnification. And don't want to lose marks for silly little things like not underlining the title, so whack a line underneath there, and that's about it. Cool.